Welcome to the Deep Dive, where we sift through the latest insights and uncover those illuminating nuggets of knowledge for you. Sometimes understanding the science behind something doesn't diminish its wonder, it actually deepens it.
Oh, absolutely.
Think about the intricate physics that make a diamond sparkle, or how birds truly achieve flight. Those sights remain absolutely entrancing. And the same goes for our topic today. We're embarking on an incredible deep dive into one of humanity's biggest questions: how life began on Earth.
A small topic, then.
Just a small one. Our mission is to trace the surprising, almost unbelievable journey from lifeless liquid to the living organisms we see today. We'll be drawing on the latest hypotheses from geochemists, particularly a fascinating article by Michael Russell, an American scientist, which explores exactly how those initial chaotic conditions set the stage for life.
It's a great piece.
So let's explore this remarkable path.
It's quite astonishing how the Earth itself acts as a massive engine for this entire process. The same force that drives Yellowstone's old faithful thermal convection is literally the planet's internal power source.
Right. Convection, like in a boiling pot, but on a planetary scale.
Exactly. We're talking about swirling currents of magma deep underground, along with water and air, which drive everything from volcanic eruptions to the mixing of vital nutrients in our oceans, and even the trade winds that crisscross the globe.
And what's crucial about this continuous planetary churning is that it constantly creates fresh surfaces within the Earth's crust, surfaces brimming with chemical potential.
That's the key phrase, chemical potential.
Imagine about 4 billion years ago, just a few 100 million years after Earth had finished forming, this constant renewal enabled some truly pivotal chemical reactions to begin.
Precisely. Picture the early ocean environment. Warm springs bubbling up from deep below the surface, bringing the Earth's internal heat and dissolved minerals to fertilize the brand new seas with a steady supply of ions from the underlying rock. These conditions, combined with energy from the sun, were absolutely essential and set the perfect stage for life's very first stirrings.
So when we look at the core chemical building blocks of early life, we're talking about hydrogen, H2, and carbon dioxide, CO2.
These basics.
These were incredibly abundant on early Earth, and even today they form the absolute foundation of all food chains. But here's the interesting paradox. These two molecules are surprisingly stable in each other's presence.
Right, they don't just react.
They don't just spontaneously react, despite holding immense chemical potential.
This begs a fundamental question. If they don't react on their own, how did life even get started? The pivotal insight is that life is both the catalyst for and the product of their reaction.
Like a feedback loop.
Think of it like a biological feedback loop. Life creates the conditions for more life right from the very start. Beyond H2 and CO2, early life also needed other essential ingredients. Nitrogen, typically as ammonia, sulfides, phosphates, and then trace amounts of specific transition metals like iron, nickel, manganese, cobalt, and zinc. All these elements were readily available in the early ocean, especially near deep-sea hydrothermal vents.
Right, those vents are crucial. And what's truly remarkable is that the very first organisms, likely what we call acetogens, which produce acetic acid or acetate,
Yeah, acetate producers.
probably emerged because the formation of acetate released a significant amount of energy. Methanogens, which produce methane, were also early forms.
Also very early, yes.
It's fascinating that even the simplest life forms produce an astonishing amount of waste.
Yes, waste is relative, isn't it?
In fact, this waste, which eventually included the oxygen that transformed our planet, is perhaps the most likely signal we could detect to indicate the presence of life on other planets.
It certainly gives you a sense of the scale and impact of life even at its inception. But to truly understand how life began, we have to grasp the absolutely chaotic conditions of the early Earth.
Yeah, it wasn't exactly peaceful.
Not at all.
Yeah.
Our planet was spinning far faster then, with days lasting only 4 or 5 hours.
Wow, 4 or 5 hours.
The moon was significantly closer, causing powerful frequent tides and storms. And giant meteorites were constantly pelting the planet, literally vaporizing the ocean surface on impact,
Just boiling the oceans away.
and throwing up dust clouds that could block sunlight for extended periods.
Given all that incredibly destructive chaos you just described, it's hard to imagine anything surviving, let alone starting. So, where did scientists propose life could possibly have found a foothold under such conditions?
It seems impossible on the surface, right?
You'd think nowhere would be safe. Yet the answer, as proposed by Michael Russell and Alan Hall back in the late 1980s, is surprisingly specific. The warm alkaline springs found deep at the bottom of the acidic Hadean ocean floor.
Deep down, protected.
This wasn't just a safe haven. It was a place of constancy and nourishment, completely protected from destructive UV radiation, never too hot or too cold, and with a perfectly stable pH.
That constancy was absolutely key. The natural gradients of temperature and pH around these hydrothermal vents provided a continuous source of energy and opportunity.
Gradients are always important in biology.
Always. The dissolved minerals constantly flowing from these springs created what essentially acted as a continuously regulated flow reactor. As these dissolved compounds met the cooler, acidic seawater, they would precipitate out of solution.
Making solids.
Exactly, forming towering mounds of carbonates, silicates, and iron-nickel sulfides around the springs, like mineral chimneys.
And this alkaline spring theory got a massive boost in 2000, right, with the Lost City discovery?
Yes. Right. This is a stark contrast to the extremely hot black smokers, which, while important sources of metals and phosphates, would absolutely incinerate organic polymers, making them unlikely sites for life's initial emergence.
So the temperature had to be just right. Not too hot, not too cold.
The Goldilocks zone, but for hydrothermal vents.
The very germ of this alkaline spring hypothesis actually came from a charming personal anecdote involving Michael Russell's son.
Oh, the chemical garden story.
Yes. He was entertaining his boy Andy with one of those chemical gardens kits, you know, the ones where minerals magically grow into colorful structures.
I remember those, yeah.
The next evening, much to Michael's dismay, Andy started breaking them apart. And from behind a locked bathroom door, Andy shouted, "Hey, Dad, these things are hollow."
Huh. Out of the mouths of babes.
Exactly.
That simple observation, surprisingly, sparks a major scientific insight. Russell and Hall realized that 350-million-year-old columns, chimneys, and bubbles found in rocks from an Irish lead-zinc mine made of iron sulfide must have been natural chemical gardens. These structures could have acted as inorganic membranes for the first cells, forming precisely when warm, alkaline spring water met acidic seawater.
So, these iron sulfide precipitates formed a gel with tiny pores and bubbles, providing perfectly enclosed sites for chemical reactions, like little compartments.
Little inorganic compartments, yes. The somewhat flimsy membranes within these structures could trap and concentrate organic molecules, encouraging more complex reactions to occur.
It's a fundamental mechanism that persists even today, as all autonomous life forms use a membrane to contain their contents for very similar reasons. Containment is key.
Absolutely. And crucially, these early protocells would have experienced a constant stream of H+ ions, protons, crossing the membrane from their acidic surroundings.
Protons, so like a tiny electrical current.
Essentially, yes. This natural energetic current is known as the proton motive force, and it still does useful work for living cells today. It's a fundamental energy source.
So life was tapping into this natural energy flow.
Exactly. Interestingly, while these early cells benefited from naturally occurring gradients, modern cells have evolved to actively create their own, pumping out H+ ions to generate this force on demand.
Wow. Okay. Furthermore, laboratory experiments showed these iron sulfide membranes were semi-permeable.
Right, selectively permeable.
Meaning they could trap larger, sticky organic products like amino, fatty, and nucleic acids and sugars, while allowing small, unreactive ones, such as acetic acid and methane, to escape.
Letting the waste out, keeping the good stuff in.
But this still leaves us with a fundamental question. How were these complex organic molecules even made in the first place? As we've noted, carbon dioxide and hydrogen are quite stable.
Maybe to push.
They needed a kickstart, a catalyst.
Exactly. So what was that first catalyst? One strong candidate is the mineral greigite, an iron-nickel sulfide. Cut598A8.
Greigite, okay.
The key insight here is the nickel it contains. It's incredibly effective at driving those hydrogen and carbon dioxide reactions, which otherwise wouldn't happen spontaneously. Nickel's a good catalyst.
And this next connection is quite remarkable. If greigite was indeed one of the first catalysts for life, then some remnant of this earliest process might still exist in life's modern catalysts, our enzymes.
Echoes of the past in our own biochemistry.
Researchers like Richard 𝗏𝗏 and 𝗏𝗏𝗧𝗏 suggested back in the 1960s that the oldest proteins contain cubanes, which are 4Fe4S centers used for electron transfer.
Iron-sulfur clusters, yep. Very ancient.
As Nobel Prize-winning biochemist Albert Szent-Györgyi starkly put it, life is nothing but an electron looking for a place to rest. That's a powerful image, isn't it?
It really is.
And in this context, carbon dioxide was waiting to be that resting place.
And lab breakthroughs truly support this idea.
Yeah.
In 1999, researchers demonstrated that enzymes containing iron-nickel sulfides could reduce CO2 and catalyze acetate formation,
Wow.
with their active centers strikingly resembling 𝗲𝘃. This wasn't definitive proof, but it was a powerful suggestion.
A strong correlation.
And earlier, in 1997, other studies showed that precipitated nickel and iron sulfides could produce acetate in lab conditions, though they used carbon monoxide, not diopside, in that specific experiment.
Okay, slightly different, but still showing the catalytic potential. So we have the catalysts, and then we have the building blocks. In the early 1990s, researchers successfully used iron sulfides and clays as catalysts to generate simple amino acids like glycine.
Glycine, the simplest amino acid. Makes sense it would appear early.
And even more compelling, a decade later, other studies demonstrated the generation of strings of amino acids, or peptides, using similar metal sulfide catalysts.
So not just the building blocks, but starting to link them together. That's a huge step.
Absolutely.
The one keystone still missing in this grand hypothesis is a proven inorganic means of directly reducing CO2 to Cl, carbon monoxide. That initial reduction step is still debated.
Still a piece of the puzzle to find.
But the overall picture is incredibly compelling. It suggests that over time, these simple peptides could have spontaneously engulfed nearby iron-sulfur clusters to form primitive enzymes, much like the ferredoxin we see today.
So the proteins start doing the catalytic work.
Exactly. And because peptides are generally insoluble, these proteinaceous films could have eventually taken over the role of cell walls and membranes.
Replacing the inorganic mineral walls.
Precisely. This is how globules of self-replicating molecules could have finally floated free of the hydrothermal mounds that spawned them. The first independent cells, maybe.
As the chemistry shifted from purely inorganic to more organic, early metabolism would have become increasingly efficient. But for Darwinian evolution to truly kick in,
Right, you need inheritance.
there needed to be some form of natural selection, which implies a rudimentary form of genetic control.
This leads us to the crucial question of information storage. Genetic information is stored in RNA and DNA. The consensus is that RNA, or ribonucleic acid, likely came first,
The RNA world hypothesis.
preceding DNA in genetic information storage. Its components were readily available in the early Earth environment: phosphate from hydrothermal vents, and the sugar ribose and nitrogenous bases like adenine from primitive materials.
Okay, the ingredients were there. But intriguingly, one RNA monomer is pretty useless as a blueprint on its own.
Right, a single letter doesn't tell you much.
So RNA must have initially had a different advantageous role in the not-quite-living cell before it became the director of the show, before it started coding for us.
And that initial role was probably as adenosine triphosphate, or ATP,
The energy
the
currency.
universal energy currency in living cells. ATP is indispensable. We actually generate more than half our body weight of it every single day,
That's staggering.
isn't it? When cells break its high-energy phosphate bonds to power themselves, the resulting adenosine diphosphate, ADP, or monophosphate, AMP, can then link up to form an RNA polymer.
So energy first, then information storage using the leftovers.
Kinda, yeah. The energy molecule itself provides the building blocks for the information molecule. It's quite elegant.
And it gets even more specific. One fascinating idea proposed by Anthony R. Marsh is that a bound chain of RNA could have actually gripped amino acids and helped form peptide bonds.
Acting like a primitive ribosome, almost.
What's so compelling here is the crude specificity. Early RNA sequences could select for certain amino acid side chains, a view echoed by Carl R. Woese back in 1967.
This remarkable specificity still exists in modern organisms. For instance, RNA codons with a central uracil tend to attract hydrophobic amino acids.
Water-fearing ones.
Right, while those with a central adenine favor hydrophilic ones, water-loving.
Okay.
Polyadenosine, for example, still selects lysine today, and the 𝗏𝗚 codon still encodes lysine in our own genetic code. It's an incredible ancient legacy.
Wow, that connection is still there. Beyond just selecting amino acids, there's the idea that nucleotides might have actually begun to assemble them.
Not just picking them out, but linking them up.
The models propose that a mineral, perhaps a sulfide, provided a foundation for an RNA template that either selected or even generated amino acids and then assembled them into peptides.
These short proteins would accumulate in the semi-permeable bubbles, with this early division of labor representing an irreversible first step toward evolution in the protocell.
The first specialization.
You could say that. The RNA world hypothesis does have conditions, though. RNA is fragile, suggesting life likely didn't start above, say, 50 degrees Celsius.
Cooler than those lost city vents, maybe.
Potentially, or in cooler parts of the system. And eventually, the more robust DNA took over many of RNA's functions as life moved into more varied environments. DNA is just much more stable.
So, given all this, what does this mean for the very first evolutionary fork in the road? While acetate generation was productive, other ways of combining CO2 and H2, like methane synthesis, offered even greater energy release.
More bang for your buck, metabolically speaking.
This led to the first major split in the evolutionary tree, proto-acetogens and proto-methanogens, which then specialized into what we know today as bacteria and archaea.
The two great domains of prokaryotic life.
So how did these early life forms survive and spread? The early ocean was essentially a desert, an ultra-dilute broth constantly irradiated with damaging ultraviolet light.
Not a friendly place.
Organisms that were blasted into the open ocean from the hydrothermal vents would have died quickly without their hydrogen source. The only safe route was down.
Down into the crust.
Into the warm sediments and permeable basalts of the ocean floor, forming the deep biosphere.
A vast, hidden realm of life that's still thriving today.
And over millions of years, Earth's mantle convection and tectonic plate movements slowly brought parts of this deep biosphere to coastal shallows.
Plate tectonics playing a role again.
Here, some bacteria found an optimal position, deep enough to be protected from harmful solar rays, but shallow enough to use longer wavelength radiation for even more organic synthesis.
Finding that sweet spot again.
This leads us to the second extraordinary event in Earth's history. Oxygenic photosynthesis.
Game changer. A huge one.
This transformed the planet, changing its face from a blue-green cast. Unlike early life's reliance on Earth's deep hydrogen, photosynthesis uses solar energy to extract hydrogen from water.
Splitting water, H2O.
Reacting it with atmospheric CO2 and critically releasing oxygen as waste.
The oxygen that we're breathing right now.
Exactly. And just like the first emergence of life, photosynthesis likely started with a mineral catalyst. This time, a manganese calcium oxide complex.
Another mineral connection.
What's so compelling here is that manganese atoms are more resistant to radiation than iron, possibly acting as a defense against destructive UV rays for early organisms.
A shield initially.
Before being co-opted to play the lead role in the process of photosynthesis itself.
That makes sense. Evolution repurposing things.
Specific minerals like ronsiate and hollandite are thought to be structurally similar to the oxygen evolving center, OEC, of photosynthesis, the very heart of the process.
The core machinery.
The fascinating part is that molecular clusters of either are remarkably interchangeable. This suggests that a ronsiate cluster near a bacterium could have morphed into a hollandite-like shape, becoming much more efficient when engulfed in a membrane. This led directly to the development of cyanobacteria.
The first oxygen producers on a massive scale.
And then when these were engulfed by other cells to form chloroplasts, this precise mechanism and the manganese calcium center were perpetuated into green plants where they remain virtually unchanged even today.
It's incredible, that same core structure doing the same job for billions of years inside every green leaf.
It's truly incredible, isn't it, how life has adapted to exploit nearly every energy source available on the planet. And what's wild is that the fundamental basics of metabolism haven't really changed in over 3.8 billion years.
The core pathways are ancient.
Even though the presence of oxygen over the past 2 billion years has allowed cells to achieve increasingly complex functions.
Oxygen provided a huge energy boost, enabling complexity.
We humans are just one result of this incredible, sprawling journey, different from other life forms, perhaps only in our unique capacity for self-awareness.
Our semi-reflective consciousness, as the article puts it.
Understanding how life started and how it fundamentally works certainly prods us to realize our responsibility for good housekeeping on this planet.
You could hope so.
Yet, here's the rub, the paradox at the heart of it all. Life's very imperative encourages us to be profligate and make waste-consuming resources as fast as we can is, in a sense, natural to our existence.
It's built into the metabolic drive, isn't it? Grow, reproduce, consume.
This raises an important question. Maybe a little less mystery and a little more wisdom about the inherent nature of life might better prepare us for what is to come. What stands out to you about this paradox?